Welcome back to KSR Biology. So in previous class we studied about glycolysis. At the end of the glycolysis, how many molecules of pyruvic acid is formed? 2 molecules of pyruvic acid is formed, right? Yes. But so what is the fate of this pyruvate or pyruvic acid? What is the fate of pyruvic acid? Yes, the pyruvic acid is the key product of glycolysis. We know that. So at the end of glycolysis, how many molecules? 2 molecules of pyruvate, and this pyruvic acid is the key product of which one? Glycolysis. Yes, what is this fate? What is the fate of this pyruvic acid? See here, so there are 3 major ways in which one? Different cells need or different cells handle the pyruvic acid produced by glycolysis, right? So what are those different ways, or what are those 3 ways in which pyruvic acid, right, so handle the cells in different ways produced by glycolysis? See here, the first one is alcohol fermentation, and the second one is lactic acid fermentation, and the third one is aerobic respiration. Which one? So alcohol fermentation, second one is lactic acid fermentation, and the third one is aerobic respiration. So alcohol fermentation, lactic acid fermentation, and aerobic respiration. So alcohol fermentation, now we will start. Yes, what is this fermentation? What is this fermentation? Where it takes place? See, already we studied about the glycolysis. So we know at the end, one glucose molecule undergo degradation or breakdown. As a result, it produces 2 molecules of pyruvic acid, and this pyruvic acid undergo fermentation. Yes. Sir, what is this fermentation? Fermentation is the degradation of organic substances by the microbes. Which one? So fermentation is the degradation of the organic substances by microbes to provide chemical energy in a reaction without taking the molecular oxygen, or without taking molecular oxygen. That is called fermentation. Alright. But who observed this fermentation process first time? The fermentation process was first observed by Louis Pasteur in anaerobic process. Who observed? So Louis Pasteur first observed the fermentation in anaerobic process. But the fermentation process was first demonstrated by, or fermentation process was first discovered by Gay-Lussac, right, ma? So the fermentation is carried out in where? So Kohane vessels. Fermentation experiment is carried out in Kohane vessels. Who first observed? Louis Pasteur. Who discovered this fermentation? Gay-Lussac. So who conducted? Where we conducted? So the fermentation process is carried out in Gay-Lussac vessels. I hope you are clear. But whatever it may be, different microbes, right, ma, carried out to different fermentation process. But among all these fermentation process, so which one, ma? So alcohol fermentation is the most common type of fermentation is alcohol fermentation, right? But wherever in alcoholic fermentation, where it takes place? Alcoholic fermentation takes place in all prokaryotic cells, and at the same time unicellular eukaryotic cells carried out this fermentation process. Yes, the first we will discuss about which one? Alcoholic fermentation. Yes, see, in alcohol fermentation is carried out by yeast, or alcohol fermentation takes place in the presence of yeast. Here, so the glucose do not undergo complete oxidation. Which one? Glucose do not completely oxidized in fermentation. Right. See, the glucose first converted into which one? Pyruvic acid. The pyruvic acid converted into 2 molecules of ethyl alcohol and 2 molecules of carbon dioxide during alcohol fermentation. The alcohol fermentation will complete within 2 steps. The first one is decarboxylation and the second one is reduction. Now we will see what is this decarboxylation, how it will take place. See, the 2 molecules of pyruvic acid undergo decarboxylation. Undergo which one, ma? So decarboxylation in the presence of an enzyme, pyruvate decarboxylase enzyme. Which one? Pyruvate decarboxylase enzyme. Pyruvate decarboxylase enzyme. So as a result, it releases 2 carbon dioxide and it also produces acetaldehyde. Which one? Acetaldehyde. Right? So the 2 molecules of pyruvic acid undergo decarboxylation in the presence of an enzyme, pyruvate decarboxylase. As a result, it produces 2 molecules of which one? Acetaldehyde. Right? These 2 molecules of acetaldehyde undergo reduction in the presence of an enzyme, alcohol dehydrogenase enzyme. Which enzyme? Alcohol dehydrogenase enzyme. Alcohol dehydrogenase enzyme. Sir, here who are acting as a reducing agent? See, in this process, whatever the NADH2s are formed in the glycolysis, these NADH2s are acting as a reducing agent. Here, the NADH2s are reoxidized and they converted into 2 molecules of ethyl alcohol and 2 NAD+. Which one? So whatever the NADH2s are formed in glycolysis, they are reoxidized. The reoxidized. So 2 NADH plus H plus reoxidized and then they converted into NAD plus and they produce which one? So 2 molecules of C2H5OH. This reaction is catalyzed by an enzyme, alcohol dehydrogenase. So what I told, the pyruvic acid converted into 2 molecules of ethyl alcohol and 2 carbon dioxide. Got it? See. So when pyruvic acid is converted into ethyl alcohol, 2 molecules of ethyl alcohol and 2 CO2. How many reactions? 2. What are those 2? One is decarboxylation and the second one is reduction. So during reduction process, NADH2s are reoxidized and produce 2 molecules of NAD plus and 2 molecules of ethyl alcohol or C2H5OH. This reaction is catalyzed by an enzyme, alcohol dehydrogenase. I hope you are clear with alcohol fermentation. But I want to mention one thing here right now. So yeast is poisonous themselves. Yeast is poisonous themselves and their death when the concentration of the alcohol percentage reaches 13%. Whenever the alcohol concentration is reaches 13, at the time yeast death themselves. I hope you are clear. This is about alcohol fermentation. Right? The next second phase, which one? Lactic acid fermentation. What will happen during lactic acid fermentation? See, in lactic acid fermentation, the pyruvic acid, which one? The 2 molecules of pyruvic acid undergo oxidation by intaking 2 molecules of NADH2. The 2 molecules of pyruvic acid, right now, so reoxidized by intaking 2 molecules of NADH2 so that it produces 2 molecules of NAD plus and 2 molecules, right now, so lactic acid. Lactic acid. This reaction is catalyzed by an enzyme, lactate dehydrogenase. Which enzyme? Lactate dehydrogenase enzyme. Lactate dehydrogenase enzyme. Sir, this lactic acid fermentation takes place in only microbes or it takes place in higher organisms? Yes. So the lactic acid fermentation takes place even in our body cells. When we do continuous exercise at the time in our body, muscle cells, so they are going to face inadequate or insufficient amount of the oxygen at the time Right, ma? The pyruvic acid undergo lactic acid fermentation. Where it takes place? Even in our body. When we do vigorous exercise, at the time availability of the oxygen to our muscle cells is less, so that the pyruvic acid get converted to lactic acid. Here, which one, ma? So 2 molecules of NADH2 is get reoxidized. As a result, it produces 2 molecules of NAD+. Whether it is lactic acid fermentation or alcoholic fermentation, whatever it may be, the liberation of the energy is very, very less when compared to aerobic respiration. Liberation of energy is very less when compared to aerobic respiration. So, I hope you are clear, but I have some small doubt. So, what is the net gain of ATPs in fermentation? The net gain of ATP in fermentation is 2 ATPs. What is the net gain of ATP? The net gain of ATP in fermentation is only 2 ATPs. Sir, how it is only 2 ATPs? Because we know that already we studied glycolysis, reaction number 7 and 10. Right, ma? 2 substrate level phosphorylations. 2 substrate level phosphorylations is equal to 4 ATPs. But in the reaction number 1 and 3, we are going to use 2 ATPs. So net gain is 2. But how many NADH2s are formed in glycolysis? 2. So whatever the NADH2s are formed in glycolysis, those NADH2s are reoxidized in where? Lactic acid fermentation and alcoholic fermentation, so that the net gain of ATPs is very less, so that the liberation of the energy in the form of chemical, means in the form of ATP, also very less in fermentation. I think it is nearly 7%. Nearly how much now? 7 or less than 7%. This is about fermentation. And the third one is aerobic respiration. Fate of pyruvate. So first alcoholic fermentation, lactic acid fermentation, and aerobic respiration. So now we will discuss about aerobic respiration. We will discuss about aerobic respiration in next video, because we will discuss in next video. Thank you.